Yo, 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 welcome to topic 3, the replication of DNA. So, the model of DNA suggested the basic mechanisms of replication. In DNA replication, the parent molecule unwinds, breaking their hydrogen bonds, and the two new daughter strands are built based on base pairing rules. So the model that was previously described is called the semi-conservative model of replication, where each daughter molecule will have one old strand, and then that's where the conserved comes into play, and then one newly made strand. So there were competing models of this conservative model and dispersive model, and in the end, Meselson and Stahl won out with their experiment. In their experiment, DNA was radiolabeled with a heavy isotope of nitrogen 15. It was placed in a medium containing the normal lighter isotope of nitrogen 14 and allowed to replicate. So the new DNA molecules that were made during this replication would use the lighter nitrogen isotope. So after one replication cycle, the DNA molecules contain half the heavy and then half the light isotope. And then this supports the semi-conservative model after the second replication, where half the DNA molecules contain only the light isotope, whereas the other half still contain the mixture of the 50% light, 50% heavy isotope. So that's what's seen here. Initially, all the DNA was labeled with the nitrogen 15, the heavy isotope, added to a medium of the lighter isotope and allowed to grow for 4 generations. So after analysis, you can see the trend that was described previously. After the first generation, we have the old and new, and then we have 50% of it being new, 50% being the mixture, and again the majority of it new, and again some with the mixture, and then the majority of it new, and then some with the mixture after the fourth generation. 88% of it being the lighter nitrogen isotope. So this is another way of looking at what was shown on the previous slide, but all in all, the big takeaway point is semi-conservative model of replication. So let's start with DNA replication. Replication begins at a special site called the origins of replication, or ORI, where the two DNA strands are separated, opening up like a bubble. And you can see that here in number 1. A eukaryotic chromosome may have hundreds or even thousands of origins of replication. And at this origin of replication, we have the replication fork. You may not see the fork until I draw it. So we see it here at the ends. Okay, it looks like a V, but the Y gives us the direction. The Y gives us the direction. I'm going to draw it over here so we can see it again. So again, the Y tells us the direction of the replication fork. Okay. Replication proceeds in both directions as seen until each origin are met with the others, and we have an entire molecule that is copied as seen in 3. So the process of replication involves over dozens of different proteins. Most of the proteins involved in replication are bound together, forming a large protein complex as seen over here. Again, the major enzymes you will need to know are these 5. Okay, we have learned these 4 before, and now we'll touch upon topoisomerase. At the origin of replication or replication fork, we have helicase. Helicase unzips the DNA, okay? And then that unzipping causes twisting and straining ahead of the replication fork as seen here. So we have the replication fork, the direction is going this way. We have topoisomerase in front of that replication fork, relieving the strain. Okay, so topoisomerase relieves the strain of the unwinding of the DNA helix. So once the DNA is unzipped, right, by helicase, some other enzymes come to play. So DNA polymerase, specifically DNA polymerase 3 in bacteria, goes in and attached to a primer and starts adding nucleotides. Okay, so DNA polymerase, once bound to the template strand, reads the template in the 3' to 5' direction and add nucleotides to the 3' end of the um, primer and continues to add in that direction. So the new strand of DNA is being built in the 5' to 3' direction. So the directionality of DNA synthesis causes the two template strands to be copied in opposite directions. So the template strand that creates a continuous strand is called the leading strand. So this newly synthesized DNA is called the leading strand because it is moving in the direction of the replication fork, as well as being one continuous strand of nucleotides being added on. So again, leading strand is the new strand that is made continuously in the direction of the replication fork. And then as the replication fork is moving along, helicase is unzipping it. DNA strand is also being created on the opposite end. So remember, DNA polymerase still reads from 3' to 5'. And this right here, y'all, this is the lagging strand. So the new strand that is being made in fragments going away from the replication fork. This is discontinuous and in fragments. So these fragments have a name. The name is Okazaki fragments. And that's the only time I will say, or sorry, not only time, but one of the rare times I would say the scientist's name. And in order to make it look like the continuous, there's an enzyme, DNA ligase, that glues these fragments together. So again, as stated before, DNA polymerase must bind to a primer. So it is the starter strand, and usually it is synthesized by a primase. So DNA polymerase adds nucleotides to the 3' end of the primer and continues until the replication of that section is completed. Important thing to note here, y'all, only one primer is needed to synthesize the leading strand. So again, that one continuous strand has one primer at the very beginning, whereas there are multiple primers set for the Okazaki fragments. Okay, so another type of DNA polymerase, and this one would be DNA polymerase 1 in prokaryotes, goes in and remove the primers, right? They are RNA, and they replace it with DNA. So, DNA polymerases can make mistakes during replication, but most errors are repaired. So, who repairs them? DNA polymerase, of course. So, DNA polymerase has a proofreading function, and if a base is paired incorrectly, the nucleotide is removed and then the proper is placed. Now, after replication, There is a mismatch repair process in which other proteins, and you won't need to know it for AP Bio, but other proteins scan for mismatch bases and then replace them with the correct ones. Well, and that is it for today. Have a good one.